Hello बच्चों, let's see the question. So the question says electroporation procedure involves. So इन चारों ऑप्शन में से हमें वो स्टेटमेंट आइडेंटिफाई करना है जो electroporation को बेस्ट डिस्क्राइब कर रहा है. So the key concept in this question is to understand what is electroporation. So electroporation is a method which is used for direct gene transfer. So direct gene transfer is a method in which we can transfer DNA or gene of our interest into a target cell without the use of any vector. So we can directly transfer the DNA into the host cell or the target cell into which we want to insert the gene of our interest. So in electroporation, high voltage of current is used for this form of gene transfer. So what is the purpose of this high voltage? So this high voltage current results in the formation of transient pores in the cell membrane of the target cell. So the cell into which we want to insert the gene of our interest, if we treat that cell with a high voltage current, then that will lead to the formation of pores in the cell membrane. So the pores which are formed in the cell membrane will now allow the entry of DNA into the cytoplasm of the target cell. So in this way, we can ensure the entry of the nucleic acid or the gene of interest into the host or the target cell. So electroporation is a more specific method of gene transfer where the gene is directly transferred into the target cell by the formation of pores in the cell membrane, which helps in the entry or the easy entry of the DNA into the cytoplasm. So now if we see the options given, so the first option says that fast passage of food through sieve pores in phloem elements with the help of electric stimulation. So this is not the correct statement about electroporation, because in electroporation, food is not passed, but instead gene transfer is done. The second option says opening of stomatal pores during night by artificial light. So again, this is not correct because a stomata is nowhere related with the electroporation method and it's opening or closing with the help of artificial light. The third statement says making transient pores in the cell membrane to introduce gene constructs. So this is the correct statement because we are making transient pores with the help of electroporation in the cell membrane of the target cell, so that that ensures the transfer or the entry of the gene into the host or the target cell. Now, the fourth option says that purification of saline water with the help of membrane system. So this is the incorrect statement also, because this is actually reverse osmosis and it does not involve electroporation in any possible way. So the final answer for our question here is option 3, that is making transient pores in the cell membrane to introduce gene constructs. Hope you understood this well. Best of luck. Thank you.